Good morning. And first, I want to thank the organizer, especially Halal, for inviting me. And it's been an amazing 2 days so far. So this was a title suggested by Halal, so I put it up there. And it's very close to what I actually do. And this is represent what my lab and as a PhD student and postdoc I've done in past over 10 years. And basically, basically we study the role of peripheral and central nervous system immune cells. And I'm an immunologist, and the reason I put this in red, I'm also located in a department of microbiology and immunology where everybody studies T cells and infectious diseases and wondering why I study brain. At the same time, before anything, I want to actually acknowledge my people who funded this work, especially Michael J. Fox Foundation, took a chance with a new PI, especially when you talk about system biology, everybody says there is no hypothesis. And I just want to show that, no, we have a lot of hypotheses, and this is the area, maybe an interesting area for NSF to actually fund it, because we will learn a lot by looking at this, not as a reductionist, but as a system biology, looking as immune cells as a whole, not only in central nervous system, but also peripheral system. So I started with microglia. This came up a few times during these 2 days. But what is an immunologist's perspective about microglia? This is one of the least understood immune cells population if you're talking to an immunologist. And when you open a textbook, it always says homeostasis, but what does that mean really? We have in microglia some, I wish this ribbon wasn't here, but there is another function there, clearing up apoptotic neuron. And then these are actually the non-immune function of microglia, something very unique to these immune cells. And again, you're talking about an immunologist studying this. They help myelin formation. They are important in synaptic remodeling. And then we have these functions that are immune function of them: antigen presentation, cytokine production. And all of these together are the attribute of these cells. These cells have been, since 2014, put in a spotlight in neurodegeneration. When I was a PhD student around 2006, we didn't have our conferences even. I was in an immunology and stem cell biology lab, and I would go to those conferences. Nobody cared about microglia. But then all these genes attributed to Alzheimer's was discovered in these cells, and they all of a sudden became very hard to study. But what is their role? And what is their immune role? How are they contributing to neurodegeneration? Is it excessive inflammation, or is it the insufficient immune response that is the reason these cells are important to neurodegeneration? But one of the fundamental questions was that, is it microglia or blood-derived macrophages? And I take you back to 2007, because these cells, still to this date, and I'm going to emphasize on this, we don't have a marker that under every condition, not only healthy, also disease, can separate microglia from blood-derived macrophages. So up to 2007, we believe that myeloid cells in central nervous system or CNS parenchyma, they are microglia, which are tissue-resident macrophages, like macrophages in many other tissues. And they come from blood-derived macrophages, which maintain this population. This was based on the phagocytic theory put forward by Cohen and Van Voorst that was saying from bone marrow, we have monocyte come into blood vessels and then go to every tissue and make them tissue-resident macrophages. However, in 2007, and as I said, before 2007, this is where we were. We used bone marrow transplant and irradiation That was the paradigm. Everybody wanted to study these cells and trace them. That's what they were using. And the field believed that they come from the bone marrow and blood. We published a paper in 2007 along with Marco Print's lab. I used parabiosis model, I wish this pointer would work, that share blood circulation without having irradiation. And Marco's lab covered the head of these mice and did the transplant. And we showed that actually in healthy and neurodegenerative model, as an example was ALS, microglia do not come from the blood, and they self-renew. They only depend on themselves to maintain themselves. But what about other models? So follow-up to that study, the question was, what about a model like multiple sclerosis, another spectrum of the neurological disorders, when we know there is inflammation, when T cells are the driver, what happens there? Again, I used the parabiosis model, and in this model, we showed that T cells initiate the disease. However, the progress depends on myeloid cells entering the CNS. And I'm going to emphasize here, because when we block this infiltration, even though microglia were activated, the mice did not get very sick. They stayed in a very early stage when T cells are required. Another observation in this paper, which is mostly ignored, is the fact that when the cells were coming in monocyte, they were leaving. They weren't becoming part of the brain. And only if there was a hematopoietic stem cell transplant, something that has a progenitor would become part of this. So the whole study, and why am I telling you about something 10 years ago, is that now I am very hesitant to call them neuroinflammation or neurodegeneration, because we need to actually define this neuroinflammation. We have diseases like ALS, that is players are microglia and astrocytes, and then bottom of here, you see Huntington disease as well. I'm sorry, this ribbon is covering it. And then we have a disease like MS, that we have cells like T cells, B cells that are actual players. So these are two different spectrums, two very separate, two different polarity of neurological disorders. And then in between, we have Alzheimer and Parkinson's that has both important component. It's not like a mess that the T cells are starting, or at least we don't know, but both cells are important. And then therefore, neuroinflammation cannot be an all-purpose definition. And why I go over and over on this, I was trained as a postdoc by Larry Steinman. Larry is an immunologist, and he studied MS and discovered Tysabri that blocks T-cell infiltration to the CNS. So I was the only microglia person in the lab, and every time I said inflammation, he was like, you can't say that unless you define it for me. And this is where we are. T-cell field is very well understood what is happening, B cells, but we don't, and we need to get there. So this is what we study. So our lab is very interested to understand the immune response and how they play a role in disease. We started in September 2019. In a disease like ALS that I showed in 2007, players are microglia. We are interested to know how microglia are responsible for certain motor neuron being resistant or vulnerable to the disease, something that is still in this disease happens and we cannot explain it. In a disease like Alzheimer and Parkinson, what we are interested is, and Malu gave a really nice talk yesterday, we know peripheral inflammation is important. How peripheral inflammation direct microglia to become neurotoxic and neurodegenerative? How is this communication is happening? So I wasn't sure if I'm going to get through all my slides, so I thought I put a teaser. So this is what working in ALS. Basically, in ALS, even the cases like mutation of SOD, when there is the ubiquitous mutation everywhere, There are motor neurons that are degenerating, all of these, but also there are resistant motor neurons, such as Eran ocular motor neuron. These patients use tracking device to communicate. So we are interested to see if all the motor neurons have it, is it the environment that is actually protecting in some versus another? In PD and Alzheimer, we are taking a very basic immunology approach. So, yes, last night during dinner, I had a talk with Sheena, and she said, convince me microglia are important. So there you go. When I give a talk or when I give a lecture, I always say these immune cells are different. They're supposed to keep everybody quiet. If our microglia like T cells work, we would run fever all the time. This is a very fragile brain. This has been studied very well. We know that neuron and microglia communicate through certain ligands, CD200 to CD200R, CX3, CR1, CX3, CL1. These are the brakes. This keeps checks on. Something happens, and such as viruses that was talked about yesterday, a misfolded protein, we don't know. These brakes are gone. And this is not something I put forward. Hugh Perry, an amazing immunologist from UK, has actually talked about this a lot. So they become primed. For first time, they've been stimulated. But that doesn't mean they are still neurotoxic. All of you talked about these cases that you see there is plaques but no disease, or there is inflammation, no plaques, but there is AD. So there is a secondary stimulation, that is systemic inflammation, aging, we heard about that, there is senescence, and these secondary inflammation comes and actually cause microglia to become neurotoxic and therefore damage the neuron. And this is the cascade, this is the concept that we are studying in these cells, in these diseases. So how are we doing this? Neurodegenerative disease models, animal models, don't recapitulate immune system, especially peripheral immune system. Yes, the plaques and everything cause a little bit of microglia activation, but we don't know exactly if that's what's happening in disease. So we're doing a reverse engineering. You use tissues from patients. We use spatial transcriptomics and multiplex imaging codecs. We also use single-cell, both RNA and CyTOF, which is single-cell proteomics, and map these diseases in these two different compartments. And then we recapitulated in iPSCs and transplanted in mice. And there were talks to these 2 days about predictive models. From here, we are guessing that is when what is created here, because we co-cultured them with neuron and astrocyte, is this means that these mice going to be developing any motor neuron dysfunction or cognitive dysfunction. So the first part is about immunity in ALS. There have been observations that microglia are important in ALS. This is a field that very much is focused on motor neuron, but there have been few very elegant studies. There's activation in ALS mouse and also patient tissues. Then in 2006, Don Cleveland lab, they deleted the mutation of SOD from SOD mice, and the mouse lived longer. In 2016, the depletion of microglia caused that the mouse lived longer. Still, SOD is the best model because of the pathology and how we can see that pathology. So, but how do they implement this rule in ALS pathology? The language of immunologists is either cytokine, signaling pathway, or surface marker receptors. And this is what I'm interested. How is this function? How are they protecting? So, wishing always during my postdoc that microglia were like T cells. This is the beauty of T cells. We know their cytokines, we know their signaling pathway, we know all their transcription factors. And until very recently, this is what microglia was, one cell. We didn't know much. So The most functional classification we've seen is M1, M2, which is, again, it's not correct. M1, M2 put forward by Simon Gordon, and himself over and over says this was done in vitro for infectious disease. Very, very different paradigm. And this is a field in terms of microglia activation, and it always really bothers me. People see the morphology change, and they say it's activated. Unactivated cells to an immunologist means something, means there is a cytokine, means there is a receptor. You can't say a cell with that many processes if it's changing, because if you're looking at a white matter versus a gray matter of healthy mouse, again, the morphology is different. Again, there is amoeboid. So there is many, many factors that it matters to define what activation means and what functionality means. So our question was, is microglia a homogeneous cellular population? And could we distinguish this based on functional phenotypes? In 2012, when I started my postdoc at Stanford, CyTOF was new. CyTOF is a single-cell proteomics, basically flow cytometry, but because instead of having the antibodies tagged to the fluorescent, you have it to the rare metal. Instead of 15 to 17 marker, all of a sudden we had 48, 49. And if anybody tells me that they can do 17 to 30 colors flow on a brain, I want to see their compensation. This brain is autofluorescent, it bleeds everywhere. There is no way you can do that many colors in brain. Other tissues are perfect. I had techs quitting on me coming from spleen, looking at brain's compensation. So this study, we identified 3 populations. We called them ABC. We weren't very creative. Around the same time, a group found exactly the same as C population that we found with single-cell RNA and called them disease-associated microglia, but that's what we called them. I'm just going to explain the ALS data. The paper is already published. So in ALS, we saw that these populations exist, all 3 of them, and then a population where B was massively upregulated to end the stage of disease. Other models that we looked at was EAE model of MS and Huntington. We basically were trying to look at the 2 polarities. We looked at cytokine, and then it was interesting in population ABC, we found these 5 cytokines being expressed. However, this is a single cell data, and we wanted to know, is every single cell express the same cytokine? So we clustered them only on cytokine, and it was very interesting. In ALS disease, so what you see here is basically fraction of cells that express a certain cytokine in each of those populations. Let's look at population B. There is a massive subset that only expresses TNF-alpha. The dark blue is there is a percentage that doesn't express any cytokine. What was interesting were 2 cytokines, IL-10 and TGF-beta. These are anti-inflammatory cytokines, and they were present. They were present from early onset to the end stage. So what we did basically here was we showed everybody inside the single cell there is, again, subsets based on functionality. So the take-home message was, while most subsets are expressing inflammatory subset, there are 2 subsets that are anti-inflammatory based on IL-10 and TGF-beta. I'm going to take you to some immunology from your immunology book, although these are very new, so I don't know how long they've been in the textbooks, but the breakthrough in immunology was the discovery of Treg and Breg. These are the cells that control immunity, like basically, and cause homeostasis because they are suppressing excessive immune response. And they balance the immune response towards any antigen or damage. So they do this function by IL-10 and TGF-beta expression. And this was very interesting for us that we were finding microglia that have exactly the same functionality based on cytokine expression. So our hypothesis is that there is a subset of microglia similar to Treg and Breg that play an important role in maintaining CNS homeostasis and protection of neuron. As we age, remember all of these neurodegenerative diseases, even ALS, that when they have this mutation happens in later in life. As we age, as the immune system breaks, we're losing that. And those neurons that are selectively resistant could be because they are actually around those subsets. So what we did CyTOF in ALS tissue from SOD patient, and it was very interesting. Around the ocular motor neuron, we could find IL-10 expressing subset. However, there were also TNF-alpha expressing subset, but not in other area, not in lumbar or cortex. We don't see that IL-10. The issue with single cell is the fact that when you analyze them, you mix them. There is a slew of the cells. So since starting the lab, we decided to use spatial technologies. So this is what the bulk RNA look like. This is where single cell suspension look like. And this is where spatial or transcriptomic or proteomics look like. And this would give us the ability to see where these subsets are. Are they close to a neuron that is protected or no? So right now, the single cell technology is spatial transcriptomic is very new, therefore it's not single cell. So what we're doing a spatial transcriptomic and also single cell RNA-seq, and then we are developing tools that combines these two, and we are able to say exactly which cells are which. So the next step is then, based on the genes that we are identifying, we are modifying the iPSCs, and then we are culturing them with motor neuron to see if they are basically driving them to motor neuron death or protection. We are doing this in mice and human. In mice SOD, we are looking at different clinical stage of the disease. And in human, we are getting ocular motor neuron and lumbar spinal cord. Ocular motor neuron being the area that is protected, and lumbar spinal cord is the area that is the most damaged. So some data in SOD mice, which was very interesting. In onset, these are all the Venn diagram of comparing the ocular motor neuron to the lumbar. In onset, most of the expression was metabolic pathways. And remember, this is a spatial, so it's not single cell, it's a still bulk. So it's usually 2 or 3 per dots, but it still is not one cell. So metabolic pathways make sense. That's where the neurons are mostly dying. But as the disease progresses and we are in an end stage, mostly were immune inflammatory cells that were actually different. And teasing about our human data was also very interesting that we found 7 unique genes downstream of IL-10 pathway upregulated in ocular motor neuron in end stage. And in mice, sorry again for the writing down there, there were 3 genes that were actually upregulated from the IL-10 pathway in onset, and 5 genes that was in end stage. So this data right now is telling us microglia could have a role in protecting selective vulnerability of the motor neuron. And this distinct subset, similar to T and Breg, could differentially be located near vulnerable motor neuron and resistant motor neuron and protecting them. And this is where we are with this study trying to map it in iPSC. I'm going to switch quickly to AD and PD. So I explained this when we talked about priming and also the fact that priming as systemic inflammation is neurotoxic. So priming microglia, what does it mean? Aggregate. And I'm putting this on a capital and rate immune memory. In past 2 days, I heard a lot about different aggregates. One of the major receptors of these cells is pattern recognition receptor. I'm very interested to collaborate with all of you to see and understand all of these different aggregates, what sort of immune memory it creates. if the difference in priming is the reason that microglia become neurotoxic or no. And again, we are interested to know what about immune cells that are getting in, which cells, which cytokine, and what is this cascade that drives microglia to be toxic. So we need to answer all these three components to be able to define inflammation in these cells, in these models. For this, we are right now taking tissues. We're using codex and multiplex imaging and CyTOF, and to answer this question, what are the cells there? What is codex? I was co-supervised during my postdoc by Gary Nolan. This is a cancer biology cells that develops a lot of tools, including codex, which is a multiplex imaging. Right now is up to 63 different marker. Basically, each primary antibody is conjugated to an oligonucleotide, which is unique to that antibody. Then there are cycles of secondary coming that is going to conjugate to those and then annealed, and we process the images. Steve today talked about a lot about the algorithms that his lab is making. We need to remember most of the technologies in single cell we use come from cancer. Our cells are different. They have processes. When the first time I ran this on our cells, it would pick stuff that were in microglia. So we spent a year to actually develop the algorithm that understand this is microglia, or this is not if a cell is inside the blood vessels, doesn't take it as astrocytes. So these are very important as we adopt these technologies to understand neuroscience is different and the cells are different. So this is an image that to show you we can do many, many colors. This was our panel. So one of the interesting discovery was finding these T cells that are next to microglia. And these T cells, we went after them because we are interested to see how this communication happens. So this study, we have subcategorized microglia based on their different markers. And right now we have 16 subpopulation. We are able to say where in different region of the brain they are. Basically, this is the map of gray versus white matter. But as you can see, when we disperse this, we can see where they are. And then we can see how far they are from the plaques, how far they are from T cells, and how far they are from blood vessels. So, sorry, I'm just going to run through this very quickly. So this is done. Yeah, so the study basically, sorry, let's go. So what we discovered was that with a subset that we found in the blood, when we cultured them with iPSC, they drive the microglia towards very inflammatory profile, very close to PMA inomycin, which is a very, very very strong inflammatory component. And basically, now the summary is that for each of these diseases, we really need to understand what is the inflammation, and we need the technology. And in our case, we have a subset of T cells that seems to be driving microglia towards inflammation. And we still are working to understand this question mark. This is my lab, and I have very great collaborators that is giving us the samples and the founding agencies that actually took chance on a new investigator. Thank you, and sorry.
This was a great talk. Thank you very much. People have implicated TGF-beta in cell death as well. For example, we showed that in hair follicles, it induces the reduction phase. So how do you know what the role here is?
So it's a very good question, and we don't until we actually looking at that TGF-beta downstream pathway to look at it. I always call the TGF-beta and IL-6 in the brain one of those schizophrenic cytokines. We don't know until we're done. TGF-beta is important in microglia survival. There's been shown with the depletion that if we depleted microglia die, but it's one of those pathways that we need to look at.
So I have a more of a general comment for all of us. I think it'll be difficult for us to define an immune profile for Parkinson's disease specifically, because I think, first of all, it's very heterogeneous to begin with, but also during the different phases of the disease, the immune inflammatory response probably changes. So very challenging task to say this is the Parkinson profile.
I agree with you, and the PPMI study we're doing, we're looking at this some to pheno-converted versus non-pheno-converted, but that is the challenge. That is the challenge we have, especially with diseases when it goes so long, and the only thing that can help, it's the same with AD. We try to get the pure ADs, that 29%, but as soon as we get to the mix, the whole microglia profile start changing, and we can't really pinpoint it.
Yes, so one of the best application of looking at somatic mutation is to better understand some kind of cell lineage. So as I mentioned yesterday, we saw some microglia mutations from the brain also found in the blood. But I think in your slide you mentioned in your earlier work, so microglia in the brain is not coming from bone marrow anymore after development. Maybe it can change with aging, with BBB breakage, and can you comment on it, how we can interpret our data?
So as I said, still to this date, we don't have marker. So in 2016, I was on the paper published by late Ben Barres that TMEM1119 was introduced as a marker for microglia, but it's in healthy. As soon as we're looking at the EAE, for example, microglia start expressing it. And as I said, most of these genes, and this is one of the issues with the field, is that mostly neuroscience and neurobiology have studied it. For example, CD33, complement receptor, all of these are also on myeloid cells in the blood. So which one are you really looking at? But to answer your question, yes, if monocytes get to the brain and stay, because in MS they don't stay, but there have been studies where Chris Bennett showed that if we deplete the microglia and then the blood monocytes come and stay, they adopt the microglia profile, which creates another level of complexity. Because right now, we can't really understand in our human AD data, we have a CD163 subset. Is it monocyte or is it coming from the microglia or monocyte? We don't know, and this is human. We can never interpret it.
Okay.
Any other questions to any of the Michelle?
Thank you. From a drug development perspective and also otherwise, usually we always want to down-regulate neuroinflammation and reduce that kind of. But you've also mentioned that microglia have very other types of functions, and we've heard yesterday, I think from Malu or someone else, that also the immune system gets less efficient with age. And aren't we ought to look at certain other pathways within microglia that could be boosted or enhanced in order to help patients with neurodegeneration rather than always looking at bringing down neuroinflammation? What's your opinion on that?
So I think this pathway, like IL-10 and TGF-beta, like Treg pathway. For example, one of the clinical trial in the ALS patient right now, which is Treg injection, because in mice it's shown very good results. But when it always was very complex why this works. ALS, it's not a T-cell driven disease. But then from this data, we see that IL-10 expressing microglia around the neurons that are actually protected. So maybe that's why that Treg injection or transplant is actually working. So yes, finding those subsets, and it's so refined, those subsets, that it needs to be really system immunology that, okay, we have this, and as we heard, it can be TGF-beta, it's one of those schizophrenic. We need to make sure in this context it's actually protective.
Any other questions?
I'll just make a comment, since you're mentioning TGF-beta. One of the earlier researchers that was doing TGF-beta a lot was Tony Wiscore at Stanford, your colleague, and he's done some really interesting studies regarding the ability of a young mouse to rejuvenate, if you will, an older mouse, first with parabiosis, now with just plasma. So infusions of plasma from exercising mice can make older mice's brains younger. And so they're, of course, trying to find out what this is, put it in a bottle, and, you know, they have a company to do this. But I think that idea supports the notion that there's communication between the periphery and the blood, and that there might be some good things in there that we could use to do exactly what you said, kind of support, boost, rejuvenate, reboot kind of thing.
I just have to make a note that when we actually culture our iPSCs with the blood serum, it didn't drive them. It was the time we put the cells in that started driving microglia to neurotoxic, which is very interesting for us because old school immunology was all ELISA and let's see what's in the slew, but now it was just becoming very funnel to very specific cell type that expresses two cytokines at the same time.